A very warm welcome to all of you guys. So today we will going to talk about another important biosynthetic pathway where we will going to synthesize one type of biomolecules from a completely different type of biomolecule. So today we will going to talk about the glyoxylate cycle where from lipid we will going to generate carbohydrate. Although it is one pathway that take part in this long conversion, meaning that from lipid to carbohydrate we have to cross near about three biosynthetic pathway. Among them, glyoxylate cycle is one, and it is very important because without this pathway, the two pathways which are remaining cannot synthesize carbohydrate from lipid, and that is the reason why this glyoxylate cycle, which is not present in our human body, cannot convert lipid into carbohydrate. Okay, so we have to study about the glyoxylate cycle. So first we will going to start our lecture with the name the glyoxylate cycle. So why the name the glyoxylate cycle? So the glyoxylate cycle especially occurs in plant, as well as you may also see it in protist, in fungi, as well as in bacteria. And mainly in plant, it occurs in a special type of peroxisome known as glyoxisome. And from the name of glyoxisome, the name glyoxylate cycle comes. Okay. And now this glyoxisome is very important because this glyoxisome will going to carry out two important biosynthetic pathway. One is the fatty acid oxidation, which is the beta acid oxidation of the fatty acid that will gives us acetyl CoA. Now this acetyl CoA will going to do this glyoxylate cycle pathway and gives us succinate. So this glyoxisome, especially in terms of plant, is very important because remember this glyoxisome is only present in plant cell. So let's get talk about the glyoxylate cycle. So first, in the introduction, we will going to see that two molecule of acetyl CoA will be used by the glyoxylate cycle, and ultimately we will get one molecule of succinate. So how the reaction occurs? So if we talk about a broad overall equation of the glyoxylate cycle, so two molecule of acetyl CoA with the help of the reduction of NAD plus in presence of water giving us succinate, right? So now most of the enzyme used in glyoxylate cycle are as same as our TCA cycle. So although there is one step which is not present in TCA cycle, but the remaining are totally same with the TCA cycle. So if you have already studied about the TCA cycle, then understanding glyoxylate cycle will be very easy for you. So first 2 points we have already discussed. So and the third point we have just discussed, right? So now we will going to directly move on the diagram to understand what is the thing that is going on. So here I will going to use this diagram. So the entire thing will start from here. I hope you can see this diagram. So start from here. This acetyl CoA will enter in the glyoxylate cycle in the glyoxisome. We are considering that the entire thing is going on in the plant cell. So what will occur? First, this acetyl CoA will going to condense with glyoxysomal oxaloacetate in presence of enzyme citrate synthase, and we will get citrate. Okay. And now what, as we have studied in TCA cycle, here same thing will going to happen, that is isomerization of citrate in presence of the enzyme aconitase, which will give us isocitrate. Now the next step is different from the TCA cycle because in the TCA cycle there was a decarboxylation reaction after that reaction, right? But here after the isomerization of citrate we will get a reaction where isocitrate lyase takes part and cleaves isocitrate into succinate and glyoxylate. This succinate will be liberated, and and this glyoxylate cycle, with the help of some subsequent step, again regenerates oxaloacetate. So this glyoxylate will be again condensed with another acetyl CoA. That's why in the introduction I have told you that two molecule of acetyl CoA will going to take part and gives us one molecule of succinate. I hope it is now clear to you. So malate synthase enzyme will going to facilitate this reaction and giving us malate. Then oxidation of malate to oxaloacetate will occur in presence of malate dehydrogenase, and also during that time NAD plus will be reduced. Okay, and that's how glyoxysomal oxaloacetate will be regenerated. And the succinate which is released here will going to first enter the TCA cycle in mitochondria and converted into malate. Now this malate have two important fate. One is this malate will be converted into oxaloacetate in mitochondria and continuing the TCA cycle, and that's how if this malate will going to do this job, then the glyoxylate cycle will be considered as a anapleuritic process. So we have already done a lecture series on anapleuritic reaction where I have told you that these anapleuritic reactions will going to maintain the level of TCA cycle intermediate despite their anabolic characteristics, right? Because we all know that the intermediate of TCA cycle also takes part in variety of other biosynthetic pathway, but still the TCA cycle is completing, right? That means there are obviously some other biosynthetic pathway which are maintaining the level of those intermediates, right? And this glyoxylate cycle, if this malate will going to convert into oxaloacetate in the mitochondria, will be considered as this anapleuritic pathway. Okay, so it is totally depending on this malate whether or not this glyoxylate cycle will be considered as an anapleuritic reaction. So now another fate that this malate can follow is it will travel to cytosol, and where it will be converted into oxaloacetate. Now you may ask, what is the utility of converting itself into oxaloacetate? So if this malate will be converted into oxaloacetate in the cytosol, then it will get the opportunity to enter into gluconeogenesis. And we all know what is the end product of gluconeogenesis. The end product of gluconeogenesis is glucose, right? So that's how, with the help of first fatty acid oxidation, that is beta fatty acid oxidation, we are getting acetyl CoA. This acetyl CoA is entering the gluconeogenesis and gives us succinate, and this succinate will going to enter the mitochondria where it will going to take part in the TCA cycle and converts itself into malate, and then this malate is entering in the cytosol where it take part in the gluconeogenesis and ultimately giving us glucose, right? So that's how in plant, bacteria, fungi and protist from carbohydrate, from lipid, carbohydrate can be generated, right? So that's the idea. So, and again, this is the entire glyoxylate cycle. So for a quick recap, this glyoxysomal oxaloacetate will going to condensed with acetyl CoA in presence of citrate synthase and giving us citrate. Now this citrate will going to isomerize with the help of enzyme aconitase and giving us isocitrate. Now this isocitrate will be cleaved by isocitrate lyase, and succinate will be liberated and glyoxylate will be created. Again, you may also say this glyoxylate pathway as glyoxylate pathway because there is a generation of glyoxylate as an intermediate. So now this glyoxylate will going to condensed with acetyl CoA in presence of enzyme malate synthase, and we will get malate. Now this malate will be oxidized, reducing the NAD plus in presence of malate dehydrogenase, and we will get oxaloacetate. That's how oxaloacetate will be regenerated and we will get our succinate, and this succinate will go into enter the TCA cycle in the mitochondria where it will get malate, and now this malate, if we'll going to continue the TCA cycle, okay, then this glyoxylate cycle will be considered as an anaphyluritic process, and if it will going to travel into the cytosol and then convert itself into oxaloacetate, then we will get glucose, right? Not we will, but bacteria, plant or fungi will get glucose, right? So that's the idea. So that is the glyoxylate cycle. So as I have told you that plant, bacteria, fungi, protist like organism have the ability to do glyoxylate cycle, and that's why they have the ability to convert lipid into carbohydrate, but we humans cannot do, right? But still there are various important human pathogens, for example mycobacterium tuberculosis have the ability to convert lipid into carbohydrate with the help of this glyoxylate cycle. So I hope this video will be very clear to you, and it will going to help you a lot to understand the glyoxylate cycle concept. So we will going to meet with another video. So till then, bye bye.